we're going to take a closer look at the clotting cascade, also known as the coagulation cascade. This is a critical process in the body that prevents blood loss after an injury. I've created a simplified diagram to provide us with a view of how the cascade works, showing the transformation of key proteins into a stable blood clot. Before we dive into the main part of the diagram, let's briefly talk about haemostasis. Haemostasis is the process that stops bleeding when a blood vessel is damaged. It has two main stages. We've got primary haemostasis shown here on the left, where platelets stick to the damaged blood vessel and form a temporary plug to reduce blood loss. And we've got secondary haemostasis, where clotting factors activate in a cascade to create a strong fibrin mesh that reinforces the platelet plug, forming a stable clot. Within secondary haemostasis, there are two separate pathways that activate the clotting cascade. We've got the intrinsic pathway on the left in green and the extrinsic pathway on the right in blue. Both of these pathways lead to the activation of the common pathway, which is where clot formation truly begins. The intrinsic pathway is triggered when blood comes into contact with exposed collagen from a damaged blood vessel. It involves clotting factors already present in the blood, such as factors 12, 11, 9, and 8. These factors activate each other in sequence. The extrinsic pathway is triggered by tissue factor, which is factor 3, a protein released by damaged tissues. It activates factor 7, which quickly kickstarts the cascade. Both of these pathways converge at the common pathway, which you can see here in the middle of the diagram. From this point, clot formation begins with the activation of prothrombin into thrombin. Now that we've covered the basics, let's take a closer look at each step in the diagram. At the start of the common pathway, we see the enzyme prothrombin. This is an inactive enzyme in the blood. Prothrombin is also known as factor 2. It plays a vital role in the clotting process. Think of it as a sleeping molecule that only wakes up when it's needed. Prothrombin is only activated when a blood vessel is damaged, like during an injury. This happens because the body needs to tightly control clotting. If prothrombin were active all the time, clots could form in places where they aren't needed in the body, which would be dangerous. By activating it only at the site of the injury, the body stops bleeding while ensuring the rest of the blood continues to flow normally. To activate prothrombin, the body relies on a group of proteins called clotting factors, shown here in the purple box. These clotting factors work in a cascade-like process where one factor activates the next, amplifying the signal step by step to ensure a fast and efficient response. Key clotting factors include factor 2, which is prothrombin itself, as well as factor 7, factor 9, and factor 10. These factors are produced in the liver and their production depends on vitamin K. Without vitamin K, these factors wouldn't function properly and the clotting process would be impaired. This connection between clotting factors and prothrombin ensures that the process is tightly regulated and only activated when necessary. Once activated, prothrombin is converted into its active form thrombin, shown here in the green box. Thrombin is also known as activated factor 2A, written like this, factor 2 with a small letter A. So activated factor 2A, or thrombin, is the central enzyme in the clotting cascade and it drives the next key steps forward. Thrombin doesn't activate on its own, it requires help from the prothrombinase complex, which consists of 4 key elements. Activated factor 10A, activated factor 5A, calcium ions, which are also known as factor 4, and phospholipids. The activated factor 10 is the main enzyme that cuts prothrombin to turn it into thrombin. Activated factor 5 is a helper protein, a cofactor that boosts the activity of activated factor 10, making the process faster and more efficient. Calcium ions, also known as factor 4, act as a glue, allowing clotting factors to bind to surfaces and interact properly. Phospholipids are found on the surface of activated platelets and act as a platform where the complex is assembled and functions efficiently. Together, these components ensure that prothrombin is efficiently converted into thrombin. Once formed, thrombin amplifies the clotting cascade by activating other clotting factors and preparing fibrinogen for its transformation into fibrin. Next, thrombin works on fibrinogen, a soluble protein circulating in the plasma. Under the action of thrombin, fibrinogen is converted into fibrin, as shown here in the transition from the yellow box to the pink box. This step is critical because fibrin is insoluble, which means it forms solid strands. These strands weave together into a mesh, trapping platelets, red blood cells, and other components to form a stable clot. In the final step, the fibrin mesh is reinforced to make the clot stronger. This happens when we have factor 13, plus some calcium ions, which helps form cross-links within the fibrin strands. The cross-links help stabilize the clot so it can withstand the pressure of blood flow and protect the injured vessel while it heals. So let's quickly recap the key steps in the clotting cascade. Prothrombin, with the help of clotting factors and vitamin K, is activated to form thrombin. Thrombin then converts fibrinogen, a soluble protein, into fibrin, which forms the framework of the clot. This entire process is designed to prevent blood loss while providing a stable structure for the body to heal the damaged tissue. The clotting cascade is highly efficient and carefully regulated, ensuring clots form only when and where they're needed, protecting the rest of the bloodstream from unnecessary clotting. Before we wrap up, let's take a moment to understand why the clotting cascade is so important in medicine and healthcare. When this process doesn't work as it should, it can lead to serious health issues. So let's look at the clinical relevance. If the cascade is too slow or impaired, as in conditions like haemophilia, the blood doesn't clot properly. This can result in excessive bleeding, even from minor injuries, and makes it difficult for the body to heal. So our example would be haemophilia. On the other hand, if the cascade is overactive, it can cause unnecessary clot formation. This can lead to dangerous conditions like deep vein thrombosis or DVT, where clots form in the veins, or strokes, where clots block blood flow in the brain. This is why understanding the clotting cascade is so important in the medical and pharmaceutical fields. By studying it, we can develop treatments to help manage clotting disorders, like using clotting factor replacements for haemophilia or anticoagulant medications like warfarin to prevent excessive clotting in conditions like DVT or atrial fibrillation. Thanks for watching. I hope this explanation has helped you better understand the clotting cascade and how it plays such a vital role in our bodies. If you have any questions or want to dive deeper into the intrinsic and extrinsic pathways of clotting, feel free to leave a comment below. Don't forget to like this video if you found it helpful, subscribe for more educational content, and share it with anyone who might benefit. I'll see you in the next video.